Welcome back. In class we left off by looking at this image, and we did number 27 together, which is asking you to label the parts of the HIV virus, which is right here. The viral envelope is the coating that surrounds the viral genome, and keep in mind the capsid encloses the DNA, or in this case RNA, and we also have some reverse transcriptase in HIV. So HIV should be a virus that you're fairly familiar with. You should have heard about it. And you should know that it infects humans, primarily. That's its host. And it is, I'm looking at number 28 now, it is a provirus. What a provirus is it's a virus that once it gets into a cell, it becomes a permanent resident, and it stays there. It does this by getting its genome into the host cell, and then as we can see right here, we have chromosomal DNA, and then we have the viral DNA right here, which is getting put in. We are actually putting the viral DNA, the viral genome, and we're inserting it into a chromosome. So every time this cell goes through mitosis, or cellular reproduction, and it makes a copy of the DNA, which was DNA replication, it's copying the viral genome as it copies the rest of the chromosome. It doesn't go away. It's a permanent fixture within the genome. So that's a provirus. Prophages, on the other hand, those infect not humans. Those infect bacterial cells only, and they do not have RNA. They have a DNA genome. So moving on to question 29, which is asking you to identify the stages of this, and I think you had actually labeled this image in class as well. I have those stages numbered here 1 through 9. This can be found on page 389 of the textbook. Although the explanations of steps might be a little bit lengthier than what I'm going to verbally explain, but if you're looking for the explanations in writing, they're on page 389. My steps, I do consolidate a few of these steps in a few different places, which you might notice. So you can either get them from me or you can get them from the book. If you get them from the book, it's going to be a little bit longer. The book actually has 10 steps, whereas I'm giving you 9. So the first step of the infection of HIV in a cell goes as follows. Step 1, what we see right here, we have an envelope with glycoproteins which enable the virus to bind to specific receptors on, in the case of HIV, the host cell would be white blood cells. So we can see that going on kind of right here. We have the viral envelope. There are receptors on the membrane of the white blood cells allowing this virus to bind to and release its genome into that host cell, which is actually step 2, the fusion part and the release of the viral proteins and the viral RNA, which we can see here. They're floating around. The viral genome is the red little squigglies, and then the proteins are the purple dots. Once the viral genome gets into the cell, we have the viral RNA right here. It's single-stranded. We have step 3, reverse transcriptase come into play, and it will use the viral RNA to create a strand of DNA. So we kind of see that viral RNA right here and the DNA produced as a result of the reverse transcriptase in blue, whereas the RNA is in red. So step 4, we see a double-stranded DNA now, whereas before we only saw a single strand of RNA bound to a single strand of DNA. So how do the viral cells get a double strand of DNA? Reverse transcriptase, once again, uses this time the blue strand from right here to create a complementary strand right here. So reverse transcriptase actually does a couple things. It copies the RNA strand into a DNA strand, and then it copies that copied DNA strand into a second complementary DNA strand. So that's step 4, is reverse transcriptase making a second copy of that DNA. Step 5, we have the DNA now being inserted into a chromosome, the DNA of a chromosome, as a provirus. So that's step 5, the insertion of the DNA into a chromosome as a provirus. Step 6, we can use the DNA and we can transcribe the proviral genes, proviral coming from the provirus, so we can make mRNA from it. This leads into step 7, which, as you should know, mRNA, once it is transcribed, it is translated. So step 7 is the translation of this mRNA strand, which creates more capsid proteins, reverse transcriptase, glycoproteins, and we see some of those going over here and here. Step 8 is to reassemble the viral capsid, which is this purple shell right here. These proteins are produced from the viral mRNA, which comes from the provirus that's inserted into the chromosomal DNA. And once our capsid is completed and closed with the reverse transcriptase in the genome, it can go to the cell membrane and it can bud off as a new virus. Notice that the viral envelope obtains its proteins from the cell itself. This is not coming from the viral mRNA. The capsid does, but the envelope does not. The envelope comes from the host cell. We can see that going on right here. So those are the 9 steps that I kind of trimmed down from 10 on page 389. And we can see in this image right here, which is actually on our next slide, a closer look at this happening in actuality. Not necessarily the insertion of the provirus, but at least the HIV virus going into the membrane of a white blood cell, and also a new HIV virus budding out from a white blood cell. Moving on, question number 30 is asking about the evolution of viruses, and you're given a box there regarding plasmids and transposons and a description of the mobile genetic element. So that's what you're looking for as we go through this. Once again, to recap, viruses, they're not living. They don't fit our definition of living things because living things can reproduce on their own, whereas viruses are obligated to a host. Since viruses can reproduce only within cells, they probably evolved as bits of cellular nucleic acid, which could have come from a variety of sources, including plasmids, circular DNA, transposons, and other mobile elements. And keep in mind, viruses are mobile, so it's probably why we're going with these as potential sources of viral evolution. Plasmids, they're small, circular DNA molecules. They're found in bacteria and other unicellular eukaryotes, like yeast. Yeast is a eukaryotic cell, it has a nucleus, but it's unicellular. The plasmids, they do exist separately and apart from the genome, and they replicate on their own. So they don't necessarily replicate when the cell goes through mitosis. They replicate independently on their own. And description of the transposons, they are DNA segments that can move from one location to another within the cell's genome, but they are part of the cell's genome. They just can go from here to there to over there to back to here, pretty much wherever they want. They're mobile, and they're DNA segments that can do that. Now, through your pandemic video that you've been kind of researching on, I heard a lot of you talking in class about how you found different things that affect crops or livestock. Some of you guys were looking at cow disease, and also humans. So diseases that are not, they affect everything that's alive pretty much, more or less. There's some other things that we're going to be talking about called viroids and prions, which can cause disease in plants and animals as well. So looking at number 31, specifically viral diseases in animals, the question is asking us for ways that viruses make us ill and something about a recovery from a cold but not from polio. Our ability to recover depends on the target cells of the virus. And I had mentioned the cold in class previously. So for example, using that cold, the target cells are the respiratory tract, and the respiratory tract are capable of repairing themselves. So we can get rid of the cold because those target cells are capable of repairing themselves. Polio, on the other hand, infects mature nerve cells, which are not capable of repairing themselves. Keep in mind, nerve cells, they kind of stay in the G0 state of the cell cycle. They don't go into the S phase and make copy of the DNA, or into the G2 or into the M phase where they create two new cells. They just simply don't divide, which is why it's so damaging when we have injury to our spinal cord or our nervous system. It's damaged, can't really be repaired. So going back to the first part of question 31, where are 3 ways that viruses make us ill? That's spelled out for you right in words on our screen here. Viruses can damage or kill cells by releasing enzymes from lysosomes to hydrolyze the cell or break it apart. Second thing, some viruses cause infective cells to produce toxins that lead to diseases, and others have an envelope that contain proteins that are toxic to the cell. So question 32, now that we know how we can get sick, question 32 is asking us, how can we prevent this? And vaccines are something that you're definitely familiar with. They're harmless derivatives of the virus or bacteria, depending on the problem itself. We're talking about viruses. But you're given a weakened form of the disease, which our immune system can overcome to build up our immune system and have a healthy white blood cell count that could attack certain viruses if they happen to try to get into our bodies. One thing that you should definitely take careful note on for your own health reasons, probably more so than this class, is that antibiotics do not cure viruses. They're ineffective. Antibiotics are only useful for bacterial infections, which is why you might get them after you have your wisdom teeth pulled or something. You'll probably be given antibiotics. But if you get a virus, the antibiotics, they can't kill that virus. Now, looking down here, antiviral drugs can help treat, though not cure, viral infections. I would imagine that this would be kind of the case that has been going on with Magic Johnson. And I know I mentioned him in class as well, where he had the HIV virus, and he went public with that back in 1991, which is kind of amazing that, 25 years later he's still living, even though that the HIV virus incubates for about 10, 11 years before you develop AIDS and then ultimately die from it. So whatever kind of antiviral drugs that he's been getting treated with, it's definitely working for him. However, that's not exactly a affordable method for, I would imagine, most people. We have a little bit left to do with chapter 19 here, 19.3. Looking at our ticking clock, I'm going to wrap this one up, and I'll pick up with question 33 on our next video.